Since the origins of life, humankind has felt attracted to the sky and asked itself questions. With the development of knowledge and through science and technology, humans have been adapting to their environment and molding it in order to improve their living conditions. The space age began when the first satellite was launched in 1957. Then, some orbital stations and different missions came, until some years later, humankind reached the moon. This milestone inspired many generations and proved that thinking about distant horizons was beneficial for the Earth. Now, we have moved on to Mars with the successful last missions. One of these, Perseverance, surprised us with this code message on its parachute. They're mighty things, so we want to join it by contributing to spatial exploration through synthetic biology. We are Navarra BioGalaxy 3, the third high school team from Navarra which participates in IGEM. Let's see a little bit of our history in order to explain the evolution of our project. It all started in 2018 with the production and purification of recombinant proteins on the International Space Station in an easy and cost-effective way. In 2019, the next team moved forward to Mars, wondering how plants could survive its harsh conditions, such as gravity, acid pH soils, and high levels of radiation. In this edition, our team continues with the work done by our previous teammates. Of the 3 research fields mentioned, we decided to focus on radiation, and therefore, that is why our project's name is aRADIAMARS. Specifically, we aim to propose a strategy to boost plants' natural protection from radiation. When we began investigating and documented ourselves, we discovered radiation was one of the biggest challenges when thinking about exploring Mars, and we felt the need to contribute by proposing a UV protection project for plants on Mars. Before we start, let's get to know the context. Mars is farther than Earth from the Sun. This may make us think that radiation is lower on Mars and that it is not a problem. But it's not that simple. The Earth is protected from radiation through its atmosphere and its magnetic field. But the atmosphere of Mars is 100 times thinner than Earth's, and it also has no magnetic field to protect it. Our mission is called aRADIAMARS, and we ask ourselves how plants could become more resistant to radiation. But, first of all, how do plants withstand radiation naturally? Exposure to UV radiation makes plants adapt to the new condition by changing their conformation. Plants effectively adapt through 2 main responses, morphological and physiological. Significant morphological changes are a decrease in the height, the reduction of the leaf area exposed to radiation, and the curling of its leaves. Besides these changes, there are some physiological responses involved. There is an epidermal and leaf thickening, a redistribution of chlorophylls in the middle of the leaf, and an accumulation of pigments capable of attenuating radiation in different cell compartments and tissue structures. These pigments, including carotenoids, flavonoids, anthocyanins, and phenylpropanoid compounds, form part of the natural protective mechanisms plants use to defend themselves when absorbing luminous energy. After researching the natural response of plants to radiation, we decided to focus our investigation on photoprotective pigments. More precisely, we wonder if an increase in the amount of carotenoids and anthocyanins would result in increased UV protection. Carotenoid pigments capture light energy that then is transferred to chlorophylls to be transformed during photosynthesis. Besides, anthocyanins are intracellular polar pigments that protect plants against UV light due to their antioxidant properties that avoid the production of free radicals. Here is when we start thinking about how we can boost the plant's natural photoprotective response. Therefore, we have set different fields of research. To start with, we want to verify if the increase of pigments protects from UV, leading us to characterize Arabidopsis plants overexpressing PAP1 and Arabidopsis plants exposed to volatile compounds. Simultaneously, we use synthetic biology to engineer multi-resistant plants to UV production Arabidopsis plants overexpressing PAP1 and UVR8 genes. In the first field of research, we used the last team's biobrick which contained the Arabidopsis PAP1 gene, because they didn't have time to prove its effectiveness. Our first objective was to characterize the plants modified with the Arabidopsis PAP1 gene, to prove if they were more resistant to UV radiation. Arabidopsis PAP1 gene codes for a transcriptional factor involved in pigment production. The overexpression of this gene aims to increase phenylpropanoid and flavonoid biosynthesis pathways that stimulate the production of anthocyanins in vegetative tissues. As a result, the transgenic plant has a darker color due to the accumulation of photoprotective pigments. In the second field of research, we studied the effect of volatile compounds on plants' resistance to UV. But you must be thinking, what are volatile compounds? Volatile compounds are metabolites produced by some microorganisms, in this case, Alternaria alternata fungi, that influence plants' metabolism, considerably increasing their growth in addition to an increase in the accumulation of photoprotective pigments. In the first part of the pictogram, we can see a wild-type Arabidopsis thaliana just placed near the fungi. Moving on to the next part, we can see how Alternaria alternata is emitting volatile compounds. In this final part, as mentioned before, when the plant detects the presence of these volatile compounds from these fungi, it starts growing faster in order to produce seeds to assure its descendants. And as we can see there, the plant not only grows bigger, also has a darker color due to the increase of the levels of photoprotective pigments. So, we grew our plants for 3 weeks, and in the 4th week, we put them in the presence of these compounds. Here, we can see our plants. The ones exposed to volatile compounds grew more and have a darker color in comparison to the ones that had not been exposed to the fungal volatiles. Once the plants used in these 2 fields of research were ready, we exposed them to UVB for 2 hours and a half to prove if an increase in photoprotective pigments means extra-ultraviolet resistance. To determine the levels of photoprotective pigments, we followed the anthocyanins and carotenoids measurement protocol. Firstly, we collect the leaves 2 days after UV exposure, and we ground them until making green dust. Then, we extract the pigments through suitable processes, and we measured absorbance at different wavelengths with a spectrophotometer. Once we obtained the sample's absorbances, we proceeded to analyze the results. Through different formulas based on linear calibration curve, we transformed these absorbances in carotenoids and anthocyanins concentrations in milligram per liter. In order to describe the plant's response after being irradiated, we represented the results as a fold change measure. To start with the results of plants overexpressing PAP1, we have these 2 graphics, one representing the changes in carotenoids and the other one of anthocyanins of wild-type and PAP1 plants after exposure to UV. We can see an increase in the levels of photoprotective pigments in plants overexpressing PAP1 in comparison with wild-type after exposure to UVB light. We observe a 38% increase in carotenoids, as you can see in the left graphic, and a 151% increase in anthocyanins, represented in the right one. In a visual exam made after the exposure to UVB, we can clearly see that the plant overexpressing the PAP1 gene suffered less damage, has a darker green color, and its leaves haven't fallen down. This way, we can confirm our hypothesis. A higher accumulation of photoprotective pigments results in higher protection of the plants to UVB light. Moving into the results of plants exposed to volatile compounds, we have represented them in these two graphics. As the previous ones, one showing carotenoids and the other one anthocyanins. In plants exposed to volatile compounds, carotenoids levels have increased 38% and anthocyanins levels 40% in comparison with the wild-type plants. In the visual exam, the most meaningful change can be observed in the rosettes, being the exposed to volatile compounds less damaged than the non-exposed plant. Considering these observations and the levels of pigments, we can conclude that the plants exposed to volatile compounds have greater protection to UVB exposure.
As we've proven that plants overexpressing PAP1 give a better response to UVB than wild-type plants, our last field of research is based on the construction of a multigenic biobrick, introducing another gene in addition to the PAP1 gene, aRAD. We aim to boost even more the plant's natural photoprotective response to UV radiation. Therefore, we built up a construction joining the Arabidopsis PAP1 and aRAD genes. As mentioned before, we already had the construction containing PAP1, so we created the one containing aRAD, formed by the constitutive 35S promoter, which makes our transcriptional unit get expressed in all tissues. The Arabidopsis aRAD gene is an ultraviolet B resistant gene acting as a receptor of UVB light and the notch terminator that stops transcription. Now, you might be wondering, why have we introduced this gene? The absorption of radiation rays by aRAD induces morphological responses that enable the plant to acclimatize and survive. But the main objective when introducing this gene is to initiate another biological pathway in addition to the one started by PAP1, increasing even more the levels of photoprotective pigments. Lastly, we joined the two constructions and our final plasmid had both genes, the Arabidopsis PAP1 gene and the Arabidopsis aRAD gene. This biobrick was formed in an easy way thanks to the golden brain method, which enables our module to be shared between the scientific community for future projects. Once our construction was correctly built, after checking everything was in order, we proceeded to introduce it into Agrobacterium tumefaciens. After that, we introduced our plant in a suspension of these bacteria using the floral dip method. This way, the bacteria was put in contact with the female gametes and therefore we obtain heterozygous transformed seeds. We think it has been made clear that our main inspiration is Mars, but we don't want to forget the Earth. That's why we've also thought about possible applications of our project here. We thought about numerous options, but when we participated in BioSpain, we realized that its foremost application was in agriculture. Concretely, the improvement of agriculture in the regions of the Earth more exposed to UV radiation. It is a fact that the population is growing, and that humans are in the need of food. It has also been documented that in places with high UV radiation, crops productivity falls. So, it could be feasible to use our modified plants which are more resistant to UV radiation to help those places and solve an urgent and present problem in thousands of people's lives. Now, we want to show you how it has been to work during this pandemic year. Our team, as well as all the previous high school team from Navarra, is formed by students from different school centers. This is part of the essence of our team, because we didn't know each other before the start of the project. During the forming process, we have suffered several lockdowns, and although we have been able to do online sessions, we rather work face-to-face. Our formation has been divided into different phases. First, we attended several sessions in the Planetarium of Pamplona to prepare the lab experiments. Then, we went to the lab physics lab to prepare and test the experiments that were designed by our instructors after sharing our ideas with them. In the last part of the project, we returned to the planetarium to work on the remaining part of the project that we needed to present. In August, we participated in the Ibero-American meetup. We presented our project to different university teams, and we received feedback from the judges. It was very helpful to continue working and improving our project in the following weeks. When we engage other people to aRADIAMARS, we realize that there are some stereotypes about spatial exploration. So, we decided to explain to our friends, families, and classmates how space exploration improves our lives. We designed several infographics to communicate this idea, and we talked about IGEM too. We highlighted the importance of synthetic biology in the present and future of our planet. And we celebrated an event in the planetarium. We invited the public to know about our project, we received feedback and ideas, and we enjoyed it a lot. As we study in different school centers, we presented the project in each of them in some sessions. After the presentations, plenty of our classmates were interested in IGEM and in being part of this project. So, we hope that the future of synthetic biology in Navarra is assured. Moreover, we had the opportunity to share our project and experience with the friends of IGEM EPIC, because they came to the planetarium to attend an online session of the BioSpain Congress, celebrated in Navarra in September. During this event, we were invited to the campus of Lekarov, in the north of our region, in an entrepreneurship meet. This motivated our team because we saw the future of synthetic biology and we want to be part of it. Besides, we attended some roundtables in the Baluarte Congress Center of Navarra during the presential sessions of BioSpain. It resulted interesting and helped us to decide the application of our project. To socialize the project, we have social media and also a blog. In the blog, we explain the process of our project to the general public. To reach them, apart from English, we use Spanish and Basque languages. This blog is still alive, so we will add some news in the following weeks. This has been our particular orbit around the Sun this special IGEM year. As we said at the beginning, Mars is our inspiration. And to finish, we want you to get as moved as we are with the message of Perseverance rover's parachute. We want you to dare mighty things. We think that all IGEM teams do great things when participating in IGEM, and we want to share with all of them their name in this special code. We want to thank all the people that have been part of the crew on this aRADIAMARS mission, that they're mighty things too, and have helped us to design more resistant plants to radiation. Join us, they're mighty things in IGEM.